Okay, guys, today we are going to go through lesson 2.3 dealing with carbon compounds. Now, to begin, we're going to talk a little bit about the chemistry of carbon. Okay, so what elements does carbon bond with to make up life's molecules? Now, just to kind of preamble this lesson, there are four major classes of molecules that we're going to talk about: carbohydrates, lipids, proteins, and nucleic acids. Now, carbon atoms have four valence electrons. If you recall from lesson 2.1 dealing with those, now this allows them to form strong covalent bonds with many other elements, including hydrogen, oxygen, phosphorus, sulfur, nitrogen, and others as well. Now, living organisms are made up of these molecules that consist of carbon and these other elements. So they can also bond to each other, which gives it the ability to form millions of different large and complex structures, everything from the graphite that is in your pencil to gasoline that is in your car. Okay, so we have the chemical structure of several molecules right here, methane being the simplest carbon-containing compound, all the way up to isooctane, this long branching chain structure here with 1, 2, 3, 4, 5, 6, 7, 8 different carbons right there, hence the octane. OCT stands for 8. So macromolecules. Now, what are the functions of each of the four groups of macromolecules? Well, first, let's talk about what a macromolecule is. So many of the organic compounds in living cells are called macromolecules because that literally means giant molecules. They're made up from hundreds or even thousands of smaller individual subunits that we call monomers. Now, when we take a whole bunch of monomers, like these little pieces right here, we put them together, we call that polymerization, and we form a polymer. Now, most everything that we find in our body is going to be made up of polymers. So, smaller units, monomers, join together to form polymers. The monomers in a polymer may be very different, or they may be identical. We'll see, especially with our amino acids later, that a lot of them are very similar, but they are still different in their own way. So biochemists will sort these macromolecules found in living things into groups based upon their chemical composition. Four major groups that I've already identified are carbohydrates, lipids, nucleic acids, and proteins. Now, carbohydrates, typically we call these sugars. So they are made up of carbon, hydrogen, and oxygen only. And this is usually going to be in a ratio of 1 carbon to 2 hydrogens to 1 oxygen atom. So living things will use carbohydrates as their main source of energy. The breakdown of sugars gives the cell, gives everything in our body an immediate source of energy. So plants, some animals and other organisms also use carbohydrates for structural purposes as well. So the main structure of wood is made up of really, really, really complex carbohydrates, polymers, that make up the cell wall. So many organisms store extra sugar, okay, starch, for example, as a carbohydrate. Now, starch, if you've ever boiled potatoes at home and that white foam that you see come to the top of the pot that'll spill onto the stove and you have to scrub it off with a Brillo pad, that's actually starch that's being released from the potatoes. Okay, so this is chemically similar to the starch that we use to iron our clothes as well. And it's when we take individual glucose molecules and we chain them together one after another, after another, after another, after another. So let's look at our simple sugars. We call them monosaccharides. So saccharide means sugar, mono means one. So literally one sugar. So besides glucose, monosaccharides include galactose, which is in milk, fructose, which is found in many fruits like your oranges, strawberries, things like that. So ordinary table sugar like you buy in the 5, 10, or 25-pound bag if you have a big family is a disaccharide. So it is formed when we combine one glucose molecule and one fructose molecule. So saccharide, again, sugar, di right here representing 2. So 2 sugar compound. Now, the larger molecules formed are known as our polysaccharides. So polysaccharides are typically going to be 3 or more carbohydrates bonded together. Now, many animals store excess sugar in a polysaccharide called glycogen. We, as humans, we store it in our muscles, but we only have enough for a few seconds of use before we have to start finding our energy from somewhere else. Now, plants use a slightly different one called starch that we already talked about. And they also make another important polysaccharide called cellulose. Now, cellulose will form this long, stringy material. So if any of you like celery and peanut butter, the strings that are on a celery stalk or the strings that are on a ear of corn, that is actually going to be cellulose. Now, lipids. We commonly call lipids fats. So things like lipids are made mostly from carbon and hydrogen atoms and are generally not soluble in water. Now, what we mean by soluble is if you take some cooking oil, for example, and you put it in a glass container with water in it. You can shake it up all day long, shake it nonstop, but as soon as you stop, it will begin to separate. Soluble meaning it does not dissolve. So other lipids are going to be waxes as well, just like the waxes that you burn in a candle or waxes that you may use to get your eyebrows done. The wax that they put on there and then they rip it off and you scream in pain, that's going to be based upon lipids as well. So lipids can be used to store energy. So some lipids are important parts of biological membranes and waterproof coverings. Now, the biological membrane that's most important is definitely going to be the cell membrane. Okay, waterproof coverings, we find lipids found in our skin. They help to waterproof the body to keep water from being able to pass on the outside in, and more importantly, from it being able to simply pass from the inside of our body out. Now, steroids are a special type of lipid, and they are synthesized, which means made by the body as well. And they are used as hormones, and they also help our cells talk to other cells in our body. Okay, so we have right here just this basic drawing of a lipid. So we have a glycerol head, and then we can have three what are called fatty acid chains right here. Sometimes we only have two. Now, if each carbon atom in a lipid's fatty acid chain is joined to another carbon atom by a single bond, the lipid is said to be saturated. Okay, so this single bond, just to recall, is represented by one line right here. Now, if there is at least one carbon-to-carbon double bond, the fatty acid is said to be unsaturated. Okay, so we see right here we have one carbon-to-carbon double bond right here, here, and here. So a lipid whose fatty acids contain more than one double bond are said to be polyunsaturated fats. Now, lipids that contain unsaturated fatty acids, like olive oil, they tend to be liquid at room temperature. So we see the data in the table below shows the melting point decreases as the degree of unsaturation or the number of double bonds increases. So we take stearic acid, oleic acid, linoleic, and linolenic right here. So this is melting point in degrees Celsius. Okay, so 69.6 degrees Celsius is going to be about 160 degrees Fahrenheit. Oleic is going to be 14, linoleic, and linolenic is, we see lower and lower and lower, and it has the same number of carbons, but different numbers of double bonds. So I'm going to ask a question real quick. Do you think that olive oil that we see right here, do you think that it has more or less double bonds than oleic acid? So the melting point of 14 degrees Celsius, that's going to be about 50 degrees Fahrenheit. So post in the comments below and let me know what you think the answer might be. Now, nucleic acids, these store and transmit hereditary or genetic information. This is our DNA. So nucleic acids are macromolecules that contain hydrogen, oxygen, nitrogen, carbon, and phosphorus. Nucleic acids are polymers, and the monomers that make them up are called nucleotides. Now, nucleotides are very, very similar. The only difference that we have in them are going to be the nitrogenous bases. So we see our basic structure right here. We have our 5-carbon sugar. Typically, that's going to be deoxyribose or ribose, a phosphate group, and then this nitrogenous base is going to be the difference here. Okay, so some nucleotides, including ATP, adenosine triphosphate, help us to regulate energy within the body. So individual nucleotides can join together to form a polynucleotide or a nucleic acid. Now, there are two different kinds of nucleic acids that we'll talk about are ribonucleic acid, RNA, and deoxyribonucleic acid. So they are named because they have different sugars, these 5 carbon sugars. In RNA, it's ribose. In DNA, it's deoxyribose. Now, proteins. Proteins are macromolecules that contain nitrogen as well as carbon, hydrogen, and oxygen, and sometimes a whole bunch of other stuff added onto it as well. They are polymers of amino acids. They perform many varied functions. They control the rates of reactions within the body. They help us digest food. They are the doers within our bodies. They even help fight disease. So these amino acids are compounds within an amino group represented by NH2 on one end and a carboxyl group, a carbon, 2 oxygens, and a hydrogen on the other end. So let's take a look down here at the general structure of amino acids. So we have this amino group on one side, the carboxyl group on the other. We have this lavender R right here. The R is going to be the functional group. Now, if you look at these two models right here, we have alanine and serine. So alanine and serine are identical with exception to this functional group. Okay, so with alanine, we have a simple CH3 right here. But on serine, we have a carbon bound to 2 hydrogens and then an oxygen bound to a hydrogen. Now, this is a very different molecule, and it gives it different properties. So all amino acids are identical, like I was talking about, and they are joined together through what's called a peptide bond once they are formed between the amino and carboxyl groups. When we do that, we end up producing a water molecule that is a byproduct. So there are more than 20 different amino acids that are found in nature, and this variety results in proteins being among the most diverse of macromolecules. Now, it's important to remember that just like every code of instructions, that if one step is incorrect, it can cause a very big problem. We're going to talk a lot about proteins and nucleic acids later on in the course when we go through genetics. Now, within proteins, we have 4 levels of structural organization. We have the primary structure. That's that sequence of amino acids, those 20 different ones that we just talked about. The secondary structure is when we start forming this twist-like structure that we see right here, and this is what we call the secondary structure. Then we have the tertiary structure. Now, tertiary is nothing but a fancy way of saying the 3rd level of construction, and it's how that twisty up part will coil up right here. So we have this purple part, this blue part, this other purple part, this other blue part. Now, sometimes we reach the 4th level that we call the quaternary structure, and this is when we have 2 or more tertiary proteins that act together. So this right here is actually a basic model of hemoglobin. Now, hemoglobin is responsible for carrying the oxygen in your blood. We all know that that is pretty important. So the question is, what happens if something doesn't go right in this? Well, when there is a change in one of the amino acids, we can have what is called sickle cell anemia or sickle cell disease. I'm sure that most of you have probably at least heard of that before, and it occurs when our amino acid, that primary structure, is incorrect. And when it occurs, you have a decreased ability to carry oxygen in the blood. If you have any questions, post them below in the comments, or you can send an email to mr.tucker.schs@gmail.com.